All right, well, thank you. We're going to do something a little different. I'm going to start out with a couple of comments. Then I'm going to hand it over to Samantha Joye, who's going to present some of the scientific activities and achievements from the EcoGig program. Well, the EcoGig program started many years before the Deepwater Horizon incident. Ray Highsmith, who was the director of the National Institute for Undersea Science and Technology at the University of Mississippi, started a microbial benthic observatory in the Gulf. This observatory had quite a few people from involved in the program, and it was also placed in the Gulf where there were lots of hydrates in collaboration with the Hydrate Consortium led by Bob Woolsey. This team of individuals went out to sea several times before the Deepwater Horizon incident occurred, and so when it did occur, they were all ready. They already had some sea time and went out right away and collected some of the first samples from the spill. And this provided the foundation for EcoGig, which then developed into 27 co-PIs at 17 institutions. One of the amazing things about this group is that they went to sea more than 20 times in the Gulf in the last 3 years. And most of those times, one of the EcoGig members was the PI of the expedition. Unfortunately, over a year ago, Ray Highsmith passed away. This tragic event sort of highlighted the need to have a plan in place prior to such a tragic event, as opposed to developing one on the fly. On a more positive note, one of the key aspects of this work is the involvement of the next generation of scientists. EcoGig has a lot of undergraduate students involved in the program, graduate students, and postdoctoral fellows. And over the last 3.5 years, some of these graduate students have handed in their dissertations have moved on to postdoctoral fellowships, while some of the postdocs have moved on to their first faculty job. Ray Highsmith was always a big supporter of the young scientists and trying to help them along, and it'd be great as part of the Gomery legacy to provide funding to help these individuals in this transition. Later on this week, on Thursday, there's going to be an education outreach session. I want to just highlight that because that's certainly an important part of all these programs. All these consortiums that have been up here have quite rigorous education outreach goals and activities. One of the things that we wanted to do and that we started out with was a fourth grade ecology activity. This activity focused on deep sea corals as a sentinel for ecosystem health and the design and fabrication of ROVs. This has grown into Science in the Stadium that happens prior to home football games at the University of Georgia and also at the University of Mississippi. And we've interacted with community service clubs such as the Boys and Girls Clubs of America. Just this year alone, and since January 1st, we've interacted with 700 fourth grade students in northern Mississippi. And this week, we're involved with another 350 fourth grade students in southern Mississippi at the Infinity Space Center. So over the years, we've interacted with thousands of individuals in this program. Another education outreach activity that reaches a lot of people was the research vessel Nautilus and the team with Bob Ballard and his crew helping to get the word out, reaching thousands of individuals over a period of a cruise. And Gary Finch Outdoors, we made several videos with him and aired coastally, potentially reaching millions of people that live along the coast. One of the questions with all of these outreach activities is, what's the legacy? It's pretty clear from the science programs that there's a definitive legacy in the publications and the data that are collected. But with the education outreach, what could be the legacy of some of these programs? Within EcoGig 1, one of the legacy aspects is the Seafloor Science and ROV Summer Camp that we started last year. This is a 1-week-long program for 6th to 8th graders. It's a STEM project, and it centers around technology activities underneath a scientific umbrella. And so this year, we're expanding to 4 different towns, 3 in Mississippi, and what we're trying to do is make that a sustainable program. And with that, I'll hand it off to Mandy, who's going to tell you more about some of the scientific activities and achievements of the EcoGig program.
EcoGig scales the Gulf system. We're one of the unique consortia that starts up in space using satellite remote sensing, goes down to the bottom of the seafloor doing deep seismic probing, and on the biological side, we work from single cells all the way up to the ecosystem level. As Geoff said, we are a heavy-duty seagoing consortium. I think the students and postdocs involved in EcoGig have done enough sea time by the time they're done with their degree or their postdoc to not want to go to sea anymore or to want to spend the rest of their life on a boat, one way or the other. But by going to sea and having the opportunity to interact with each other on board ship and in port, we do media events and teaching and EPO events before every cruise. We have a very strong opportunity to interact and generate really collaborative science. So this is what I'm going to highlight in the talk today. This is supposed to be the fate of Macondo oil, not the fat of Macondo oil. We had a little Mac PC transition problem, and there are lots of little things that may show up here, and really they weren't in the presentation when it started. But we're going to talk about food web incorporation. Eric touched on that a little bit. Microbial formation of marine snow, which is a part of the cross-Gomery MOSFET effort. Sedimentation of oil to the seafloor and its fate on the seafloor. The impacts of sedimented oil, and then I'm going to talk about dispersant impacts on corals and microbial populations, which I think Peter will touch on some of your comments a minute ago. In terms of productivity, we've had over 340 research contributions so far, and this is just within EcoGig 1, not counting EcoGig 2. 74 papers, I believe, published so far, 42 in review or revision, over 200 conference presentations, including 50 at this conference here. So this is a satellite remote sensing image that Ian McDonald at Florida State put together, and this shows you the footprint of the Deepwater Horizon oil spill. The red areas had a more frequent coverage of oil. The blue areas a less frequent coverage of oil, but you can see this is an extensive area that was covered by oil. The average footprint was around 22,619 meters cubed of oil. That's a big volume, and the area was around 11,800 or so square kilometers. So this was a huge area impacted by oil, and this is just surface oil. I'm not talking about the oil in the plumes or the gas in the plumes, but one of our basic questions is what happened to this material? Where did it go? Did it get incorporated into the food web? Did it get sedimented to the seafloor? Where did it end up, and what happened to it? Some of the work by Geoff Chanton is shown here. This is the Delcy-13 of particulate organic carbon versus the radiocarbon content of that carbon. And as you can see along this line, the slope of the mixing line indicates methane as a fossil carbon source to the food web during the Macondo event. This work was published back in 2012, I believe, in Environmental Research Letters, and it corroborates some of our work showing really high rates of methanotrophy early on during the spill, but that activity was not sustained over time, but it was enough to have a food web impact. Some of the other work that we've done that was mentioned earlier in Dr. Spinrad's talk is looking at the formation of microbial oil snow. It turned out that because EcoGig scientists were some of the first out in the Macondo area after the blowout, we actually saw these big giant mats of microbial oil slime all over the surface of the ocean, and then it all disappeared. It was covered and then it sank. And it was over the course of a couple of weeks where you had the formation of this material in huge amounts and then the sinking of this material. Uta Passau, Kaiser Vogel, and others have been studying the dynamics of formation, sedimentation, and impacts of marine snow, and I'm going to talk a little bit about that over the next few slides. There are many mechanisms of formation of this oil snow. One is associated with phytoplankton activity, particularly diatoms. Diatoms exposed to oil aren't very happy. They excrete slime. This slime emulsifies, attracts, sticks to oil, which can facilitate sinking. And this just shows you the percentage of oil carbon in diatom aggregates, and it can be up to 65% depending on the type of diatom. On the other panel is shown that oil is required for the formation of mucus-rich oil snow. You don't get this stuff forming naturally. It forms in response to oil presence. And if you add dispersant, you don't get the formation of this stuff either. So microbial production, whether it's phytoplankton or bacteria, microbial production of this oil snow is a natural mechanism for both facilitating biological degradation of oil and removing oil from the surface. So it acts as a very efficient dispersant, if you will. It gets the oil off the surface, but it also facilitates biodegradation, which I'll show you later that dispersants perhaps don't do quite as well. So what happened to this oil? On a cruise, one of the first, this was the 4th or 5th response cruise that we did through the EcoGig group in August and September of 2010, UTA Passau deployed a sediment trap at the spot of the yellow circle there, a little bit to the southwest of the Macondo wellhead. This was an area that was impacted by both the deepwater plume and was heavily covered by surface oil. And this is one of the other hallmarks of EcoGig is in situ instrumentation and technology. We put a lot of instruments in the water, we put a lot of instruments on the seabed, and we track in situ fate of material using sophisticated technologies, and this is just one. The image on the bottom is the collection, is a series of pictures of the collection cups from that sediment trap starting in August of 2010 and ending in October of 2011. And as you can see, stuff that sediments out of the surface ocean isn't supposed to be black. Usually it's pink. But you can see that this is heavily impacted by oil deposition well into 2011. In fact, this is a very busy figure, and I don't expect you to digest it all in the 30 or 45 seconds that it's going to be on the screen, but the sentence at the top says it all. Oil sedimentation continued for at least 6 months after the well was capped, and you can see that in the bottom plot on the lower left. That's barium and alpha olefins. Barium is a drilling mud tracker. Alpha olefins is also a drilling mud tracker. You can also track this stuff to some extent by its biomarker composition, but once you add dispersants to the mix and biological weathering, it's really hard to get a 100% fingerprint to Macondo. I remember one of the stories I always tell is I have a sample that was collected about a quarter of a mile from the wellhead in a giant thick surface slick of oil. I don't think anybody would argue that it was from the blowout, but it doesn't fingerprint to Macondo. And it's, you know, a quarter mile away. So it's very, very difficult to fingerprint this highly weathered material to Macondo, but there are a lot of other proxies that when put together can make a strong argument for this being Macondo sourced oil. So Eric showed this figure before. Geoff Chanton is both part of Deep Sea and EcoGig, and this was a collaborative project where we collected a lot of cores in this area that you can't really see the octagon. But what you should notice is the green areas to the south and southwest of the Birdsfoot Delta, the Mississippi. Those are areas that are very, very enriched in radiocarbon depleted material, i.e. oil. It's dead carbon that's on the seafloor. And what Geoff's work showed with this coring effort and mapping effort was that about 3 to 5% of the oil released from the wellhead, that's about 10 million gallons, if you're curious for a number, ended up on the seafloor. And it's very hard to constrain this number because we obviously can't core the entire Gulf of Mexico, but based on interpolation, that's the number that we come up with as a conservative estimate for how much is on the bottom. You can also use DOS, which is part of the dispersants, to look at the fate of material in the water column and track it down into the sediments. And what you see here is some data from Jennifer Field and Matt Perkins. And these top samples are samples from the Kyle traps, and the sort of orange-colored parts of the bar plots don't meet the diagnostic DOS ratio, and that just means that it could be something other than DOS, but it might be DOS. If it's green, it's definitely DOS. There are a lot of compounds that when you run it on the mass spec, it could sort of kind of be DOS, but it might not be DOS. The green stuff is definitely DOS. It makes all the diagnostic tests for DOS. And what I want to point out is that in 2010, 2012, and 2013, we're still seeing dispersant residue in the sediments. And this is nanograms per kilogram. It might sound like a lot, but given how much dispersant was released, that's a lot to be finding in sediments distributed, you know, broadly from around the Gulf. These particular samples are just representative samples from a variety of distances from the wellhead up to 30 kilometers or 30 nautical miles away. I want to switch gears now and talk about biological impacts on the seabed, and this is some of Chuck Fisher's work that that Dr. Spinrad also mentioned, tracking corals and individual branches of over time and looking at the impacts. And what Chuck's group has done, and I think it's just brilliant, is they digitally map these corals and then track, by taking photographs over time at high resolution, loss of branches over time. And Chuck just mentioned to me before that many of these corals, and you can see it in the first picture on the left, sort of see that white little blob on the middle of the coral? That's an associate that lives with these corals, and those associates, they protect the coral in a way. You see the green area in the second figure. They move around, they sort of wipe and wash and knock off the sedimented oil, and those areas tend to do better than areas that don't have those associates. And you can see that over the course of time, this is a time course going from left to right. In the final image, the blue pieces are pieces that have actually fallen off the coral over time. So the impacts of the oiling of these deep water corals, which are animals that grow very slowly and live for hundreds of years, so can't easily be replaced, those impacts are still being felt. And we don't really know the true impact on the deep water coral community yet, because we're still seeing loss of corals over time. This is a figure that Chuck made, and it just shows you sort of in a spatial sense the degree of impact and the locations of impact. The Macondo wellhead is the red star, and you can see where there are red or blue. The blues are frowny faces, reds are very unhappy faces, and then there's some green faces interspersed there where the corals weren't affected. But you can see there's a broad range of areas where corals were impacted by fallout from the Deepwater Horizon oil spill. And this work was published in PNAS last summer. So for the last couple of slides, I want to talk about impacts of dispersants, and then I'm going to end up with some physical oceanography if I have time. But in my group, we've been very, very interested in simulating the impacts of the deepwater plume in the lab. We spent a lot of time developing methods to make water-accommodated fractions of oil that were replicable so that we could add consistent amounts of carbon, whether it be oil-derived carbon, dispersant-derived carbon, or chemically enhanced, essentially dispersant plus oil. And we do these using roller tables in big bottles and triplicate, and it makes for a monster of an experiment when you add in all the controls and treatments. And I want to show you just a few results of one of these experiments that a postdoc, Sarah Klingdienst, in my group did a couple of years ago, and this paper is currently in review. So I showed a little bit of this yesterday, and I'm going to show a little bit more of it today. But if you go from left to right along this plot, you've got essentially the in situ population, which changed over the 2 weeks between collection and initiation of the experiment. You've got the first set of plots is the biotic control, which changes over 6 weeks. The second series of boxes is the oil WAF. The third is a dispersant-only treatment. The fourth is a dispersed oil treatment. And the final is a dispersed oil treatment plus nutrients. The top set of bars is cell counts. And I want to make a couple of important points. One is that when you're looking at dispersant impacts, there are two things that really, really matter. One is how you apply the dispersant, like how do you introduce the dispersant of the dispersed oil into the system. Having a consistent amount of carbon added is really important. The second is the metric that you use to evaluate what is the impact matters. Because you can see that if your metric was cell counts, then you would conclude that the dispersant alone was stimulating microbial activity more so than anything else, because that gives you the highest cell counts. But it turns out that it's much more complicated than that. And you would also conclude that in the dispersed oil treatment, you have lower cell counts, so perhaps less of an impact on the biological community. One thing I want to point out about the lower slide, which I talked about in the genomics session yesterday, we see dispersant-driven selection of microbial communities in response to both either dispersant alone or dispersed oil addition. And that dispersant-driven selection of microbial communities specifically and dramatically alters oil biodegradation, which surprised me quite a bit. And I show that here in a very busy figure, and I apologize for its busyness, but look at the first two plots. The panel on the far left is oil, is just oil. So just water accommodated fraction of oil. And the top is hexadecane degradation with a C14 tracer. The second row is naphthalene degradation. The third is bacterial production. The latter three columns are exoenzyme production. And 3 points I want to make. The highest rates of degradation of oil occur in the oil treatment, oil alone. When you give the microbes that can degrade oil, they degrade it, and they do very well at degrading it. They become nutrient limited ultimately, but for 6 weeks, they do pretty good. Corexit does not stimulate oil degradation in these experiments, and we've done subsequent experiments to repeat this one, and we've also, this is in deep water, we've also done it in surface water, we see the same thing. Corexit doesn't stimulate oil biodegradation, and that's because it selects for a microbial population that isn't good at oil degradation, it's really good at dispersant degradation. So that's a really interesting finding, and I think one that's really important and relevant for the discussion in terms of dispersant use following oil spills. We've done similar experiments with corals in Eric Cordez's group, and this is some work of a PhD student in his group, Danielle Young. And what she has shown, these are experiments done in 2012 and 2013, is that different corals respond to oil and dispersant in very different ways. Crude oil exposure is on the top, dispersant exposure is in blue in the middle, and crude oil plus dispersants is in the bottom. And what you can see is that these animals, they respond to oil much better than they respond to dispersed oil or dispersants alone. They just do much worse if there's dispersant in the mix. So again, this is very similar to what we see in our microbial experiments. You know, oil alone, the microbes are just fine. You add dispersant to the mix, whether as dispersant alone or as dispersant plus oil, and they don't do as well. Ileana Baum's group at Penn State has done some transcriptomics work looking at gene expression in response to oil in two variants of the two color morphs of the black coral, and what you see here is that these two color morphs respond very differently to oil exposure with a variety of metabolisms that I don't have time to go into, but the point is that it's very, very different. The response is different, and that's probably due to the microbiome of these different color morphs of these two different corals, and this is ongoing work that we're going to continue into EcoGig 2. The final two things that I wanted to touch on is the physical oceanography. In EcoGig, our goal is to collect field and experimental data that can be incorporated into physical oceanographic and process-oriented models to advance the understanding of the fate of oil in the environment. And this is some work from Annalisa Bracco's group that shows the importance of seasonality in where the water's flowing in the Gulf and in terms of what leads to instabilities in the water column. In the winter, it's submesoscale frontal instabilities. In the summer, it's inflow of the Mississippi River. So that means that if the Deepwater Horizon oil spill had happened in the winter versus the summer, the oil would have been distributed in a totally different way. So that's a very important finding. The other thing is the issue of scale. Physical oceanographers have models that run at a variety of different scales. Annalisa she's been working very, very hard to pull her models down to smaller and smaller scales. And the thing I want to draw your attention to in this figure is in this picture here is the figure on the bottom to the right-hand side. That's the horizontal reproducibility of the deepwater plume as a function of a 5-kilometer versus a 1.5-kilometer grid model. And you can see that these smaller grid models give you a much better representation of what that particular deepwater plume look like. So we got to bring these models down to the appropriate resolution to really capture the dynamics that were going on during the Deepwater Horizon oil spill. And so I'm going to stop there and leave this slide up for you to gaze at while I take any questions that you might have.